Okay, so today we will be talking about vitamin A. In this lecture, we will be talking about the structure, absorption, and recommended daily allowance of vitamin A. So let's start with the very basic, that are the structure of vitamin A. As you know, vitamin A is a fat-soluble vitamin. Vitamin A and its related compounds are collectively known as retinoids. Basically, retinoids are the natural and synthetic forms of vitamin A. The retinoids contain three main compounds, that are the retinol, the retinol, and the retinoic acid. We will discuss the structure of each of these compounds, and finally we have another form of vitamin A, that is the condensed form of vitamin A, that is known as the beta carotene. We will also discuss the structure of beta carotene in this video. So let's start with the structure of retinol. Retinol is the vitamin A alcohol. It means that retinol contain a hydroxyl group at its end, at the end of the side chain. Here is the structure of retinol. The retinol contain two basic parts. The first part is this ring, which is known as beta ionone ring, and it has a side chain. This side chain has two isoprenoid units. The first unit is this one, which contain four double bonds that I have mentioned with the this green lines. These are the four double bonds in the side chain. So this is one isoprenoid unit, and it has a second isoprenoid unit that contain an alcohol group, a hydroxyl group at the end. So this is the structure of retinol. When this retinol oxidizes, then it converts into another form that is known as retinol. And the only difference between retinol and retinol is that retinol contain hydroxyl group at its end, while retinol contain an aldehyde group at its end. So that is the only difference between retinol and retinol. Retinol and retinol are interconvertible. It means that retinol can be converted into retinol, while retinol can be converted into retinol. Retinol is found in animals as retinol is found in animal tissues as retinyl esters associated with the long chain fatty acids. Okay, now we will discuss the structure of retinol. As I have already mentioned, that retinol and retinol are only differentiated from each other by this group, that is the in retinol it has a hydroxyl group, while in retinol it has an aldehyde group. Now we come to the third retinoid form of vitamin A, that is known as the retinoic acid. It is also known as vitamin A acid. This retinoic acid is formed when the retinol is oxidized, and here you should know this thing, that retinol and retinoic acid are not interconvertible. It means that once retinoic acid is formed from retinol, then retinoic acid could not be converted back into retinol. The reaction is irreversible. And the difference between retinol and retinoic acid is that retinol has an aldehyde group at the end of the chain, while retinoic acid has a carboxylic group at the end of the chain. Then we come to the fourth form of vitamin D, that is also known as provitamin. Basically, the provitamins are those forms of vitamin, the provitamin A are basically those forms of vitamin A that are derived from plant sources. So these beta carotenes are also obtained from plant sources. That's why these are also known as provitamin A. The beta carotene, here is the structure of the beta carotene. Basically, the beta carotene is the condensation product of vitamin A. It means that one beta carotene contains two molecules of retinol. that is this compound, the retinol. So here it is one retinol molecule and this is another retinol molecule, and these are the side chains that are these chains, and at the end of each chain there are the aldehyde groups. So these aldehyde groups are bonded with each other with double bonds. So this is the structure of beta carotene, and when this beta carotene reaches the intestinal mucosal cells, then in the intestinal mucosal cells this beta carotene is cleaved to two compounds that are the retinol and one retinol and another retinol. Then these retinols get reduced to retinol in the intestinal mucosal cells. This retinol get reduced to the retinol form of retinoids that are the provitamins of vitamin A. After studying the structure, the basic structure of vitamin A, we will discuss the absorption of vitamin A, that how vitamin A is absorbed from the intestinal mucosal cells into the blood plasma. Alright, so after studying the molecular structure of vitamin A, we will discuss about the absorption of vitamin A from the intestinal mucosal cells, and we will also discuss the transport of vitamin A in the blood plasma. So let's start with our diet. As we consume diet that contain vitamin A from different sources, plant sources as well as animal sources. The vitamin A that we consume from plant sources are in the form of beta carotenes, while the vitamin A that we consume from animal sources are in the form of retinol esters. Then in the gastrointestinal tract, the intestinal brush border epithelial cells or pancreatic cells release a specific enzyme that is known as the hydrolysis. This specific enzyme, the hydrolysis, will hydrolyze the retinol esters into retinol and free fatty acids. The retinol ester contain long chain fatty acids, then those fatty acids will be detached from the retinol esters. So these are the detached fatty acids that are the free fatty acids, and this retinol esters is now converted into retinol. Then what happens to the beta carotenes in our GI system? So this is the intestinal mucosal cell. The beta carotenes are also hydrolyzed by a specific enzyme. That specific enzyme is the beta carotene 1515 dioxygenase. Here I have put a red star, and here also is a red star that denotes that a specific enzyme. So the beta carotene are also hydrolyzed by this specific enzyme, that is the beta carotene 1515 dioxygenase. This beta carotene is hydrolyzed to retinol, that is the aldehyde form of vitamin A, and then this retinol will get reduced to retinol, that is the alcoholic form of vitamin A. Here also that is the retinol. Then in the intestinal mucosal cell, this retinol will get esterified. It means that this retinol will get attached with the free fatty acid again, and this retinol will be converted into retinol esters again. Then these retinol esters are incorporated into chylomicrons, and then those chylomicrons move through the lymphatic system of our body. So these are the chylomicrons, and these retinol esters are incorporated in them. Re means retinol esters, and these chylomicrons along with the retinol esters move through the lymphatic system of our body, and when and from the lymphatic system these chylomicrons enter into the liver hepatocytes. Then in the liver hepatocytes, here these are the all trans retinol. What are all trans retinol? As I have already mentioned the structure of retinol previously. So all trans retinol are those form of retinols. As you know, that the retinol contain a ring and a side chain. So let's suppose this is the ring and this is the side chain. When this side chain is in straight form, then that form of retinol is known as all trans retinol, and when this side chain has a little bit bow shape, then that form of retinol is known as cis retinol, that we will discuss later on. So this all trans retinol in the liver hepatocytes will be stored in the form of retinol palmitate. It means that the storage form of vitamin A, the storage form of vitamin A is retinyl palmitate, and vitamin A stores primarily in our liver. When vitamin A is needed in our body, then this vitamin A is released from the liver in the form of retinol, and it is believed that zinc helps in the mobilization of vitamin A from the hepatocytes of our liver. Then what happens, that this all trans retinol, the vitamin A, will move to the target tissue. So let's suppose we have the target tissue, our bone. I have taken the bone as our target tissue. Then it was all about the absorption of vitamin A. Now we will discuss, after these points, we will discuss about the transport of vitamin A. Up to here, it was the absorption of vitamin A from this point up to this point. That was the absorption of vitamin A, and from here we will discuss about the transport of vitamin A. So vitamin A could not be transported freely in the blood plasma. It rather is transported by a specific protein, a specific plasma protein, that is known as RBP, retinol binding protein, RBP. So when this retinol get attached with this plasma binding protein RBP, then retinol RBP complex is formed, and this is the retinol RBP complex. Then on the surface of the target tissue, let's suppose this is our bone. So on the surface of our bone cells, there is a specific receptor for this RBP complex. Then this RBP complex will move to the peripheral sides of the cells, and this retinol, out of RBP complex, only the retinol will move into the interior of the cell, and this RBP, the retinol binding protein, will get detached from the retinol RBP complex, and this RBP will move back and will get another retinol and will transport it to the target tissue in the blood plasma. Is that clear? Now in the target tissue, let's suppose the bone, the retinol will be converted into retinol, that is the aldehyde form of vitamin A, and that retinol will be converted again into retinoic acid. So let's suppose I have mentioned the retinoic acid here with this blue mark. Then this blue mark, that is the retinoic acid or retinol, this retinoic acid will enters into the interior of the nucleus. So now here it has now entered into the interior of the nucleus. In the interior of the nucleus, there is a specific receptor that is known as transcriptional factor. This specific receptor, that is known as the transcriptional factor, this retinoic acid will get attached with this receptor, and the configuration of this receptor will be changed, and a retinol receptor complex will be formed. So this is the retinol, this blue one is the retinol or retinoic acid. So this is the retinol and this one is the receptor. So the retinol receptor complex has now been formed. Then this retinol receptor complex will move to the periphery of the DNA and will express a specific gene or will activate a specific gene. Then after the expression of that specific gene, messenger RNA will be formed, and after the formation of messenger RNA, this messenger RNA will be translated by ribosomes into a sequence of proteins. So let's suppose that protein which were synthesized here helped in the formation of bone cells, that are the osteoblasts or osteoclasts or whatsoever. So this was all about the function of vitamin A. As you know that vitamin A has many functions that I will discuss in the upcoming lectures with detail, but here you should know one function of the vitamin A, that it helps in the formation, that it helps in growth and differentiation of cells. So let's suppose this specific protein that has formed here helps in the differentiation of bone, differentiation of different cells, and those different cells in the bones are osteoclasts and osteoblasts and so on. Now here. These all trans retinol will move to the retina of the eye. In the retina of the eye, this all trans retinol will be converted into all trans retinal, that are the aldehyde forms, and then these all trans retinal will help us in the visual cycle. It means that it helps in the world's visual pathway. I will discuss it with detail in the upcoming lectures. So up to now we have discussed about the structure and absorption of vitamin A, and also the transport of vitamin A in the blood plasma. And also we have discussed these two functions of vitamin A, that it helps in the cell differentiation and growth, and also it helps in the visual cycle. Besides these two functions, vitamin A has two more functions very important, that vitamin A helps in the epithelial tissues and mucous membranes and epithelial tissues, especially is the skin, and also it helps in the reproductive system. It promotes or it enhances the spermatogenesis process in males. So up to now it was all about the structure and absorption and transport of vitamin A. After this we will discuss the RDA, that is the recommended dietary allowance of vitamin A, that how much vitamin A is recommended on daily basis to be consumed. Okay, so after studying the absorption and transport, we now will study the RDA, that is the recommended dietary allowance of vitamin A. The unit that is used for the recommended dietary allowance of vitamin A is retinol equivalent rather than international units. 1 retinol equivalent equals to 1 microgram of retinol, that equals to 6 micrograms of beta carotenes, and that equals to 12 micrograms of other carotenoids, and that equals to 3.3 international units of vitamin A activity from retinol. It means that 1 retinol equivalent equals to 3.3 international unit of vitamin A activity from retinol, and 1 retinol equivalent or 3.3 international units of vitamin A activity from retinol equals to 10 international units of vitamin A activity from beta carotenes. So this was a brief introduction to the retinol equivalent, that what is the retinol equivalent, and how much quantities it is equivalent or it is equals to. Now we will discuss the RDA for males. For males, the recommended dietary allowance of vitamin A, that is the retinol product, is vitamin A in the retinol form is recommended 1000 RE, that are the retinol equivalents, and 1000 retinol equivalents equals to 3500 international units, and 1 international unit equals to 0.3 milligrams. So 35 international units multiplied by 0.3 milligrams equals to 1050 milligrams. This quantity is for males. For females, the quantity is 750 milligrams. For females, total RE, the retinol equivalent, is recommended at 100, and that at 100 equals to 25 international units, and 1 international unit equals to 0.3 milligrams. So ultimately the at 100 RE or 25 IU will be equal to 75 milligrams of retinol daily. Here you should remember one thing very important, that this quantity, this RDA, is recommended only for retinol and not for retinoic acid. This quantity can be increased for pregnant women, and also lactating mothers. But you should remember that this quantity is only for retinol and not for retinoic acid, that I will discuss in the upcoming lectures, that retinoic acid has a very bad and negative effect on the pregnant women. Now we will discuss about the sources of vitamin A, that what are the sources of vitamin A from animal from animal sites. So the sources of vitamin A in animals are liver, fish liver oil, fish contains cords or sharks, and milk, cheese, butter, while in plant sources the green and dark, the yellowish and dark green vegetables are rich in vitamin A. That includes carrots, spinach, pumpkin and mango, etc. So up to now it was enough about the RDA, the recommended daily allowance. After this we will discuss the world's visual cycle, that is a primary function of vitamin A, and that is related to the visual cycle of our eyes. And also we will discuss in the upcoming lectures about the deficiency symptoms of vitamin A and the hypervitaminosis A, that both are the pathological conditions of vitamin A.